big hello to my wonderful PU2 warriors. I hope all of you are ready with your bows and your arrows, having just one goal in your head, and that is to top your PU2 board examination and also crack any competitive exam where your PU2 board syllabus is on priority. Well, remember, we are going to be there silently behind your back, okay, guiding you throughout the entire process, and we just want that one thing from you guys, and that is to put your trust on us. So we are trusting you with one thing, that you guys are going to be consistent in watching these videos, consistent in solving the questions, consistent in giving your best, and we just want one thing, and that is to put your trust on us. Remember the previous year also when we launched Brahmastra, we saw how humongous it was. So many children got the benefit of this particular series, my dear children, and I am not joking. So this year it is even going to be even more better, and I am telling you with utmost honesty, because yesterday also I slept at 2 a.m. Not to say that I actually started off a bit late to make this PPT, but still it went too long because I tried to incorporate all the questions that have come in the model question paper 2025-26, so you do not have to run around anywhere. So every question along with your question number is being put in this particular video, so you get a reference that where is this question taken from. Okay, and you get an idea, okay, I am covering the model question paper questions of this year as well. And not just that, there will be questions taken from the question bank, the revised question bank of this academic year, the previous year question papers and the previous year model question papers as well. So please not be worried, just put your trust, and if at all you do, please put this down in the comment section that I am in here with consistency and trust. I really want to see that. And always remember, there is a hype whenever a series is being brought, and then children thinks like, okay, I'll study this later, I'll do this later, and it goes on. No. This whole thing is planned in a way that every single day you have to invest some amount of your time with us, and with that you will be super confident to crack your examinations, my dear children. So please do that. It should not be like a one-day thing. And today I have taken the chapter Biotechnology Principles and Processes because of the reason, because this chapter is untouched. This didn't come in your midterm examination. We have to start off with new chapters as well. So it's not like we will not be covering the previous year, the midterm topics out there. We will be doing that, but it is going to be in a sandwich manner. So according to me, for biology, I will be following this pattern where one new chapter and one old chapter will be there. In that way, you can cover everything together. So I have taken Biotechnology Principles and Processes. The next lecture will be based on the midterm topics, which is of high weightage. Okay, so this chapter gives you how many marks according to the new blueprint? It gives you 7 marks. It was the same the previous year also. There is 2 1-marker questions that are going to come from this chapter, and also 1 5-marker question that is going to come from this chapter. Ma'am, can we score 2 marks and the 5 marks from this particular session? Yes, you can, if you listen to me very carefully. Okay, so are we ready? I hope we are ready. Let's get ahead with the model question paper questions 2025-2026. So the first question right here taken from MQP1, that is model question paper 1. This is question number 10. So in the year 1963, the 2 enzymes responsible for restricting the growth of bacteriophage in E. coli were isolated. One of them cut the DNA and the other added the group. What group? So ma'am, I didn't understand the question at all because I do not know the concept. Don't be worried, I'll tell you the concept also. So in the year 1963, one very important thing happened. Okay, scientists were looking forward into this E. coli bacteria. So E. coli is a bacteria. It has its own genetic material. It will be having its own genetic material. Now suppose this E. coli is being infected by a virus whose name is bacteriophage. Okay, its name is bacteriophage, and it is going to infect it. In what way does it infect? Basically, it passes down its genetic material inside the E. coli. Why? Because it wants to slowly get incorporated into its own genetic material. It's the genetic material of the E. coli, and whenever the E. coli replicates, this particular material is also going to replicate, and the proteins are going to be made in favor of this particular virus out there. But the E. coli, my dear children, on the other hand, is very clever. What did it do? It basically had two enzymes for self-defense. Okay, E. coli also has studied self-defense. What about us? Have we? Think about it. Okay, so anyway, so over here there are two enzymes. One enzyme is known as restriction enzyme. Restriction enzyme, super important. Why do you think it's restriction enzyme? Because it is going to restrict something. So basically, the restriction enzymes are the ones that are going to cut the genetic material. Okay, it's cool. It is going to cut the genetic material of the bacteriophage only or of the E. coli as well. Well, it can cut the E. coli's genetic material as well, which is why E. coli thought of another enzyme as well. Okay, which is going to add some groups right over here on the genetic material. What do you call these groups as? These groups are methyl group. These are what? Methyl group. Ma'am, why does it release this particular thing, and why there is addition of methyl group over here? Such that the restriction enzyme know that this genetic material should not be cut. So it's like a mechanism saying that whoever has the tag of methyl group should not be cut, and whoever does not have the tag should be cut. So this guy over here didn't have, which is why it is cut and cleaved, such that it cannot come and incorporate now. Okay, and this guy is saved because it had methyl group added onto it. So these were the two enzymes which was isolated in the year 1963. Okay, in E. coli. So one was restriction enzyme and the other one added methyl group. So my dear children, with respect to this concept, I will tell you one more thing. So 5 years later, 5 years means 1963 plus 5 years, 1968. Your first restriction enzyme was discovered. So isolated was different, soon studies were being made, and finally first restriction enzyme was discovered and its name was HIND2. Always remember its name is what? HIND2. Okay, so first of all this isolation was done. Second thing, that is how you can remember two. Okay, the second thing which was there was actual discovery of restriction enzyme, and that was HIND2. And my dear children, these restriction enzymes which are there, they are going to cut n number of base pairs. Okay, DNA having n number of base pairs out there. So what is the thing over here for HIND2? So we do not know the sequence. It's not given in NCERT. I'm not going to tell you that extra information because it will just make your brain work more. So I do not want to do that. Just remember, there will be 6 base pairs. So it is going to make a cut at a site which has 6 base pairs. 6 unique code is there. Okay, that is not there in NCERT. You do not have to study it. So every restriction enzyme which is there, it will have its own unique restriction sites. So this restriction site will have 6 base pairs. Please remember that as well. So in this, with the help of this particular question, you understood 2 concepts. Okay, one is this and the other one is this. Please remember. So 5 years later is being mentioned. It's not mentioned about the year. So either way you can remember it, but please remember it was HIND2 and there was 6 base pairs. Going ahead with the second question, my dear children, this is for you guys. This is not for me. This is for you to read and understand whenever you can. Please follow us on WhatsApp if at all you have not followed. And those of you watching this video without subscribing the channel, subscribe to it right away. It is going to be super helpful for you. Okay. So this notes will be given in the form of PDF through the links, okay, in the WhatsApp group. So please join the group as well. And be patient, you will get the PDF. Don't be worried if at all you do not see on the first day, you will get it, okay? Now here is, as I said, everything is happening. I have explained this to you, so which is why I don't need to explain again. It will waste a lot of your time. So let's go ahead with the next question out there. Let's see from which MCQ. Okay, so again, model question paper 1, question number 11th. So which of the following is the correct representation of primer annealing step in PCR? Primer annealing step in PCR means what? So basically, my dear children, in this whole chapter, what are we doing is that we are taking a gene of interest. For example, in my body, there is a gene that is coding for some protein, and this gene is super important. And I want to increase its production. What do I do? First of all, with the help of restriction enzymes, I am going to cut this particular gene of interest. Before that, there is a whole lot of process. Okay, I have to get into the cell and this and that. Isolation of DNA has to occur. That is the first step. Later on, I'll cut it with restriction enzyme. I'll take this. And then, my dear children, since restriction enzyme is just going to cut the DNA, okay, into multiple fragments, these fragments have to be separated with another process that is known as gel electrophoresis. Once it is separated, my dear children, I have my gene of interest. Now, I am basically going to amplify it. I am going to increase its number. Okay, I have to increase its number first of all, because I cannot just rely on this particular gene. I have to increase its number, and then I'll put it in some very important things which will do its own replication, okay, alone. That's a whole separate thing. So first of all, I just want to increase the number, and then I will put it in something else which is known as a vector, which somehow will be helping me in increasing of number later on as well. So I'm not going to that. First of all, we have to see how do you amplify the number of the DNA out there. So my dear children, you have the DNA strand like this, right? One will be from 3' to 5' end, other will be from 5' to 3' end. So first thing that I need to do is I need to separate these strands. This process is known as denaturation, where I will be separating these strands. The next step that I am going to apply is basically now I have two separated strands, one from 3' to 5' direction, another from 5' to 3' direction. Now I want to make a copy of this. I want to have replication being done. So for replication, my dear children, you always remember there is a need of an enzyme, DNA polymerase. Okay, and DNA polymerase always works from 5' to 3' direction. That means it is going to make a new strand in this direction. So over here, this is 3' to 5'. So that means new strand can be made from here to here. Can be made in 5' to 3' direction like this, opposite way. But for DNA polymerase to start off the process, it needs one very important thing which you call as primer. Okay, there is a need for primer because DNA polymerase cannot initiate by itself, which is why there is a need of primer. So the primer comes and attaches over here. Similarly, on which side is the primer going to attach over here? On this side, because again the DNA replication is going to happen like this. The DNA replication is going to happen like this from 5' to 3' direction. Similarly over here as well. Okay, so this process of attaching of primers is known as annealing. It is known as annealing. So you have denaturation, then annealing. So an annealing means just attachment of primers. Just attachment of primers. Now the next step is basically where DNA polymerase will come actually into action and it will extend this. It will be extending this. This process of extension in 5' to 3' direction is known as extension itself. It is known as extension and it is done by the DNA polymerase. Well, remember this whole process is happening at very high temperature. So the usual DNA polymerase cannot work at this temperature, which is why we take the DNA polymerase from a bacteria whose name is Thermus aquaticus. Thermus aquaticus. So this bacteria is living in a very high temperature region, which is why its DNA polymerase is also thermostable. Thermostable means it is stable at higher temperature. So I am taking the DNA polymerase of this bacteria, which is why I am going to call it TAC polymerase. Okay, TAC polymerase is what? Thermus aquaticus polymerase. That means DNA polymerase of this particular organism. Fine. Now over here you are asked which of the following is the correct representation of primer annealing step. So you have to look forward where, my dear children, one should be 3' to 5' and the primer should be attaching over here towards the 3' region. Similarly, if it is 3' to 5', here also the primer should be attaching at this particular region. So what do you think is the correct answer for this? Here it is 5'. So yes, here it is attaching properly, but on the other hand, if you see the next strand, that is also in the same polarity. Do you think that can happen? And also, here it will be 3' to 5', no? It's attaching towards the 5' end. That means, again, it will be, extension will be happening in 3' to 5' direction. That is not possible. Similarly, over here it's also, it is not possible. Why? Because both of their polarity is the same and here it's wrong. Again, here if you see, it is attaching at the 5' end. Here also it is attaching at the 5' end. Wrong. It has to attach at the 3' end. Okay, it has to attach at the 3' end. Always remember that. So what is your answer for this question? Your answer would be D. So what have you learned from over here? So DNA, DNA amplification process happens in 3 stages. One is denaturation, the second one is annealing, and the third one is extension. Who does the extension and everything? It is the DNA polymerase extracted from Thermus aquaticus, which you call as TAC polymerase. The enzyme is known as TAC polymerase because it is highly thermostable. All of this process happens at super high temperature and it can withstand that. Easy. Okay, and always remember primers attached on the 3' end and extension happens from the 5' to 3' direction. Done? Cool. So 3 steps and 3' end. You can remember like that. So your answer for this question would be D. So this is the entire process given in front of you in your NCERT. Go through it really well. I have given you the explanation as well. Next question, children, are you liking this video? Do you think I am explaining you extra concepts as well along with the questions? Then do not forget to share it with your friends and also do not forget to hit on the like button. Now the chemical nature of the primer used in DNA polymerase chain reaction. The chemical nature of the primer used in DNA polymerase chain reaction. Oh, I didn't tell you the name of this reaction. Okay, this process is known as polymerase chain reaction. And why do you need it for? First of all, you need it for amplification, as in increasing the number of DNA. So they are asking you the primers that you are attaching over here, what is its nature? What is it made up of? DNA nucleotides itself or something else? So basically always understand the primers are made up of ribonucleotides. Okay, and RNA primer is usually being used and that's how the DNA polymerase goes and does the whole thing. But in the case of polymerase chain reaction, my dear children, it is DNA primers itself that are being used. So out of this, you have ribonucleotides, you have deoxyribonucleosides, you have ribonucleosides, and you have deoxyribonucleotides. What will be your answer for this particular question? It will be D. I'll tell you why. So basically, what do you mean by deoxyribonucleic acid? So over here, this is structure of the pentose sugar. 1, 2, 3, 4, 5 carbon is there. Fifth carbon is attached to the phosphate group. First carbon is attached to the nitrogenous base. Now usually in the case of RNA, on the second position you have an OH. But in the case of DNA, this oxygen is not there, which is why now you will call this deoxyribo. Okay, this is deoxyribo sugar that you have. When it is attached to nitrogenous base alone, it's not there, just nitrogenous base is there, then you will call this nucleoside, deoxyribonucleoside. Now when you have a phosphate group also being attached, you will call this deoxyribonucleotide. So which is why your answer is not A, B, C, D. First of all, A and C is not there because it is ribonucleic acid. RNA is not involved over here. Deoxyribonucleoside is not there because over here you have the phosphate group. And these phosphate groups, my dear, each of these molecules going to come together. This phosphate group is super important for the phosphodiester group to be formed, for a primer to be formed, which is why your answer would be this. Now this question is a question number 11 itself. It's for visually challenged children of MQP1. So basically there's a replacement of the previous question. So the previous question was this, right? Replacing that question for visually challenged children, you have another question that has also been covered over here. I hope it's clear. Now going ahead with the next question. So this is how it looks. As I said, deoxyribo sugar, phosphate group and base is there. Cool. Now fourth question says that, and this is from again MQP1, question number 39. So this, my dear children, is a 5-marker question from MQP1. So I have taken all the model question paper 1 questions first, then model question paper 2, then 3, and then we'll go ahead with the question bank questions, previous year questions and things like that. Okay, first of all, we are covering the model question paper questions. Now explain the process of preparation of competent host for transformation with the recombinant DNA. What do you mean by competent host? So basically, I took the DNA, I told you, I took the gene, I basically cut it, and then I had gel electrophoresis being done, so the separation is happening, then I amplified it also, my dear children. And now basically, this amplified DNA which is there, it is being put into something. Okay, suppose there is a vector which I call as plasmid. So this plasmid is basically a double-stranded DNA structure. Okay, it's a double-stranded DNA structure which is usually there in all the bacteria. The bacteria has their own genetic material and this is an extra chromosomal material out there. It is not generally coding for something super important for the E. coli. It's just there. It's just self-replicating because it has all the tools for self-replication. For example, it has the origin of replication sequence. A sequence which is there for what you call the process of replication to happen smoothly, for process of replication to initiate off. Right? Everything is there in this particular plasmid, which is usually there in the bacteria. Now what I'm going to do is that since it has a machinery to self-replicate, I am going to insert my gene of interest over here. Okay, my gene of interest, the gene that I want to replicate is being put over here in this particular plasmid. Now, Basically, it's there, it cannot survive like that, it has to be put into a host. This particular plasmid, the recombinant plasmid which is there, it has to be put in a host, and in the host cell what will happen, my dear children, it will have everything else so that the replication can have smoothly. That process of basically putting this inside, putting this inside a cell of an organism where it can actually do the entire process is known as transformation. I am basically transforming the cell, making it get the recombinant plasmid. I am making it get the recombinant plasmid, or basically, suppose if not recombinant plasmid, I am making it get any other plasmid which is not its own. I am taking it. This process is known as transformation. I am transforming this particular cell. This is known as transformation, and the way that it can take in things is known as to make it competent. It's known as, I want to make it competent for making the entry of plasmid very, very smooth. So I have to tell you the process in which way the cells are made competent to take up the plasmids out there. I hope that's clear. It's not too much confusing, right? When I explain you the answer, it will be all the more clear. So we'll go with the A option first, and then I'll explain you the other things. So first of all, preparation of competent host for transformation. So what we have to do is, suppose we have bacterial cells out there. Bacterial cells which are there, they are treated with divalent cations. Divalent cations such as CaCl2. That calcium is a divalent cation. Now why do you do that? Basically, the Ca+, the calcium ions which are there, they neutralize the negative charges of the DNA. On the DNA is negatively charged material, you neutralize it. Why do you neutralize it? Basically, in that way, things can be easily getting inside the cell membrane. If it remains to be positively charged, my dear children, suppose I have a cell over here and the DNA along with the plasmid is right over here, my dear children, do you think it can get inside easily? No. So first of all, I have to neutralize the negative charges on the DNA. And also the mixture of cells and the DNA is then given a heat shock and cooled on ice. So basically, first of all, I treat it with calcium ions. I neutralize it. Now the next thing that I am going to do is giving a heat shock treatment. What do you mean by that? So over here I have the cells, suppose E. coli cells, and over here I have the plasmid also, which is having my gene of interest. Now basically all of them I had put in CaCl2 medium, where the Ca neutralizes every charges out there. Now the next thing that I do is give a heat shock treatment, where basically first of all I am going to keep all this thing in ice, and then later on I am going to increase the temperature. For example, I am going to increase the temperature to 42 degrees Celsius. Not increase the temperature, it can be in different plating also, but I am telling you. Anyway, the temperature over here was very cool first. Now the temperature is 42 degrees Celsius. And this is kept for how much time, my dear? 90 seconds. And then again, the temperature is made down, and it is what? Very cold. Very cold. What is the impact of doing this? Basically, this creates temporary pores. It creates temporary pores in the membrane such that it can take up this particular plasmid out there. Such that it can take this particular plasmid out there. And this process is what I say, is what I call as to make a cell competent. To make a cell capable enough to take in the recombinant DNA out there. So first of all, I hope you understood what are the things that are done. Divalent cations, these cations neutralize the negative charges on the DNA. Then both of them are kept in cold first, then in heat, then back in cold. This creates temporary pores which allows the recombinant DNA to enter the bacterial cell. So this is the preparation of competent host for transformation. Please remember, these are the steps that you need to write. So first cold, then heat, then cold. And the hotness as in 42 degrees Celsius. Now going ahead with the next part of the question, refer to the given diagram of PBr 322 and answer the question that follows. So over here there's a question that is given to, a picture that is given to you of a plasmid. The name of the plasmid is PBr 322. Now mention the restriction enzymes that can be used for insertional inactivation of antibiotic resistant genes. Too much? And how many cloning sites are present in the given PBr 322? So first of all, I'll go with the easy part of the question, that is how many cloning sites are present in the given PBr 322. So this, my dear children, I can explain to you. Next question is given now. So over here you have PBr 322, right? This is origin of replication site. as in this helps in the whole replication of this particular plasmid to happen very smoothly. ROP site is basically responsible for production of a protein that is going to help in the process of replication. This site, this gene which is there, my dear children, it's a gene, okay? It's a gene that provides tetracycline resistance. What is tetracycline? Is an antibiotic. So suppose I am growing this PBR in a tetracycline medium, it can still grow. Why? Because it is resistant to tetracycline. I have the gene for that. So that is what you mean by tetracycline resistant gene. Similarly, I have AMPR. But what do you mean by that? It is ampicillin resistant gene, another antibiotic out there. So again, if I grow this particular PBR in an ampicillin medium, do you think the cells having the PBR can grow easily? Yes, because it has the resistant gene. Now, my dear children, you know the genes, ampicillin resistant, tetracycline and ORI and ROP, everything, right? Now, here are the sites. Sites of what? The enzyme, the restriction enzymes. So first of all, I'll start with tetracycline. Over here, you have a site of SAL1. So this particular enzymes can have a cut over here. The restriction site is over here. The unique DNA site is right over here. They will make a cut over here. Suppose they make a cut over here and my new gene of interest has been put over here. Do you think this particular plasmid is still going to be tetracycline resistant? I am telling you, if I put my gene of interest over here after cutting, after cutting the segment, I'm putting my gene of interest. Do you think it will be still tetracycline resistant? No. Why? Because I made a cut over here. This gene is now not active. It is known as insertional inactivation. I inserted something, which is why it has got inactivated. Now, so that is for SAL1. Similarly, BAM-H1 site is also over here. So two sites over here. Now over here, if you see, there are two sites again for different enzyme, PST1, PVU1. Both of them 1-1. And in between, you have sites for EcoR1, CLAV1 and HIND3. This is not HIND2, this is HIND3. Now in ROP site also, you have a enzyme which is PVU2 site. Okay, PVU1 was there, PVU2 site is over here. So how many sites are these? These sites are known as cloning sites. Okay, so how many sites are there? 1, 2, 3, 4, 5, 6, 7, 8. I have 8 cloning sites. This was the question that was asked to you. That is, how many cloning sites are present in the given PBR 322? It is what? 8 cloning sites, and you have to name them. Now mention the restriction enzyme that can be used for insertional inactivation of antibiotic resistant gene. Now they are asking you, what is the restriction enzyme that I can use such that the antibiotic resistant gene, my dear children, it is inactivated now. I'm inserting something into antibiotic resistant gene and they are now inactive. So you know there are only 1, 2 and 3, 4 genes present on the antibiotic, 1, 2, 3, 4 sites of restriction enzyme that are present on the antibiotic resistant gene. So it is BAM-H1, SAL1. If I insert something with the help of this particular enzyme, if I make a cut because with the help of this particular enzyme, tetracycline resistance will not be there. Okay, similarly, PVU and PST1, if I make a cut over here, again ampicillin resistance will also be not there. I hope this is clear. Well, you have to remember the names of each of this. You would have to make a diagram of this. This diagram is super important and I'll tell you a way in which you can remember this. Okay. So we'll read the answer first. So I told you how to make a preparation of competent cells for transformation. Transformation is nothing but the process of basically inserting some another plasmid or anything out there which is new for the cell. And you're transforming the cell in a new manner. That is what you mean. Sometimes it can be recombinant DNA. Okay, sometimes it can be recombinant DNA, sometimes it would not be a recombinant, it will be a normal DNA out there also. It can happen like that. Anyway, transformation is happening since you're getting something new. Okay, now from the given diagram of PBR 322, restriction enzyme used for insertional inactivation of antibiotic resistant gene, BAM-H1, SAL1 or PVU1, not PVU1, okay, just a second. SAL1 inactivates the tetracycline resistant gene. Now PST1 and also PVU1 inactivate the ampicillin resistant gene. The number of cloning sites in PBR 322 is unique and these are the things. So how do you remember it? I'll tell you easy way of remembering it. First of all, try to make the diagram and that's the best way to remember things. Okay, so over here you have the PBR 322. Okay, here is the origin of replication, your ORI site. Here is your ROP. Okay, protein is super important for replication to happen. And here is your tetracycline resistant gene, and here is your ampicillin resistant gene. Right? Now, how many cloning sites do you have? You have 8 of them. Make 2 over here. Okay, make 2 over here. Make 2 over here. Make 3 over here. Okay, and make 1 over here. So 2, 2, 3. Okay, 2, 2, 2, 2, 3 and 1. Fine. So 1, 2, 3. You can remember like that also. Fine. Now we have to name them. So PST1, PVU1, PST1 and then PVU1. Then you have Eco R1. So you can remember this is PE period. Physical education period, PE period. Okay, so PST1, PVU1 and then you have Eco R1. So first period is PE. Now my dear children, you have CLAV1, okay, and you have HIND3. So this is C and H. This next period is what? Chemistry period. Remember, you can be asked, what is this? What is this? So you should know each specific sites. This is chemistry period. Now we have a period for college, business systems. Okay, now we have business systems. So this is BAM H1 and this is SAL1. All of them 1, 1. Okay, here, all of them are 1, 1. So BAM H1 and also SAL1. Now over here you have the cousin brother of PVU, that is PVU2. This is PVU2. I hope you will remember this. This is first is PE class, then you have chemistry class, then you have business system, then you have physics class. Okay, so 1, 2 and 3. Fine. And the third one is 3, so which is why it ends also with 3. This is how you can remember it. It's just a way. You might have different ways to remember it. You can use that. Cool? Chalo. So that's all my dear children. I'll make the changes in the PT. Do not be worried. Now going ahead with the next question out there. So this is from model question paper 2. So we have officially finished all the questions from model question paper 1. Okay. So in the given diagram of PBR 322 vector, identify the restriction sites A, B, C, D. So now you will know what is A, what is B and what is C and D. Chalo, let's do it. Let's do it. You see how super important this question is. So A is origin of replication. What is B? It is antibiotic resistant gene site. What antibiotic? Ampicillin resistant, right? Over here it will be what? Tetracycline resistant, sorry. And over here you have this site and this will be what? HIND3. The last one is what? HIND3. So A is what? A is your origin of replication and go for the last option. D is what? D is your HIND3. So I just have one option that suffices that and that is B option. And that would be B option. So origin of replication, B1 is ampicillin resistant, C1 is tetracycline resistant and D1 is this. So your answer is B for this particular question. This is again question number 11 of MQP2. Okay, here is a picture given to you. I hope it's clear. So even though I am taking time in explaining you the concepts, I think this is going to help you really well in your exam. So that is why this session might be a bit long, but my dear children, it's going to be super useful. Now, restriction site of Eco R1 enzyme is. Now that is something new, ma'am. We do not know this. So this is again this is a question for visually challenged children. I told you. So usually there's a replacement towards the end. So this is actual question. Okay, now for visually challenged children, there is another question given in your question paper itself. So this is that question, okay? So restriction site of Eco R1 enzyme. So this is an enzyme, it will have a site and it's just like HIND2. Remember the first enzyme, HIND2 is just like that. Eco R1 also has a restriction enzyme site of 6 base pairs. Again 6 base pairs. What are the base pairs? Ga, TC. Okay, if you sing a song in the class, you'll get TC. That's how you can remember it, or whatever way you want to remember, you can remember. But there are 2 A's and there are 2 T's. Always remember A and T are going to be same. Remember, we studied in molecular basis of inheritance. This has nothing to do with that. They are towards the side, but still I'm telling you. How many number of A are there? That means number of T is also there. And G and C is also there equally. Okay, you can remember like that. Fine, so 6 base pairs are there. Now, the Eco R1 which is there, it will make a cut in between G and A. It cuts between G and A. And always remember the restriction site, suppose it's over here, it's like that. Similarly, on this complementary strand, it will be like this also, GAA, TTC. So here again, they will make a cut. Okay, the enzyme makes a cut between G and A here, and on the complementary strand also it makes a cut. So the restriction site of EcoR1 enzyme is 5-GAA-TTC, 3-GAA-TTC. Ma'am, you didn't teach that. So it is 5. Okay, it is 5 and 3 dash. Always remember 5 dash, it's very unique. Usually it's not like that, right? But over here it's like that. So it is 5-GAA-TTC. Okay, cool. It's not 3, do not do that mistake ever. So this is how it is being done. I told you, no? So basically you use a restriction enzyme to cut the DNA. Similarly, with the same restriction enzyme, you're going to cut the plasmid where this DNA is going to be inserted. Why do you do that? So here I have a picture and I'll explain you that as well. This is going to be helpful for you, children. Listen to me carefully. So this is the target gene, okay? So I cut it over here. The target gene was like this. On both the sides, you had here you had GAA-TTC, here also you had GAA-TTC. You made a cut over here, which is why G and A separated. Here also you make a cut. What happens is that you get an L-shaped thing. Here what is left is an L-shaped thing. Similarly, on the other side also you make a cut. You need to make a cut both sides. That's only how you will get a target gene completely, right? So you have an L-shaped cut here, an L-shaped cut over here. See, with the same enzyme, my dear children, you are going to make a cut right over here in the plasmid also. So in the plasmid also you will have a L-shaped thing. You see, this is an L-shaped and this is an L-shaped. Now when you insert this, don't you think it will perfectly fit in? This L is gonna fit in over here and that one is gonna fit in over there. That's how you get a recombinant plasmid. And what do you call these NSAS? What do you get when you cut things with this, what do you call restriction enzymes out there? You get sticky ends. Okay, you get sticky ends. Always remember this. The sticky ends will help in basically ligating both the plasmid DNA as well as the new gene of interest. Okay, so there is an enzyme which will do the after work of it. It is known as DNA ligase. So DNA ligase is the enzyme that is being most helped by this sticky ends out there. So sticky ends makes its job really easy. It's DNA ligase, nothing else. Don't do that mistake. Cool, fine. And what do you call this? These are known as sticky ends. Now, this is a fill in the blanks question that you got in MQP, Model Question Paper 2, and the question number is 19. So fill in the blanks by choosing the appropriate words from those given in the bracket. So these are the options. Okay, you have to fill in this particular blank. The plasmid DNA acts as dash to transfer the piece of DNA attached to it. The plasmid DNA acts as what? So plasmid, which is there, okay, DNA, basically which has my gene of interest, acts as what? To transfer the piece of, it acts as what? A vector. Okay, it's a vector. Remember, vectors are carriers that carries thing. You have in mosquito also, the Anopheles mosquito is a carrier of Plasmodium. Similarly, this guy is also carrying my gene of interest, which is why it is known as vector. The plasmid DNA acts as vector to transfer the piece of DNA attached to it. Okay, so your answer for this question would be vector. Cool. Next question. Okay, I have explained you that. I don't think we need to do it again. This is a 5-marker question. This is a 5-marker question from Model Question Paper 2, and this is question number 44. Give reasons for the following. Restriction enzymes are called molecular scissors. Why do you think restriction enzymes are called molecular scissors? Because, my dear children, they are cutting molecules, big molecules like DNA, RNA, things like that. Okay, so they are known as molecular scissors because they are precisely cutting at specific sequences. You have to mention that. And you can give an example that EcoR1 cuts in the restriction site 5-GAA-TTC between G and A, and it creates sticky ends. And this is happening within the molecules such as DNA. That is why it is known as molecular scissors. We'll explain, we'll see the answer for this, okay? So if you want, we can see it right now. Because they recognize and cut DNA at specific base sequence recognition sites, just like scissors cut at precise point. Give an example, talk about EcoR1. Okay, so that it has a site GAA, TTC, 3 dash, and it makes a cut between G and A. Fine. Now next thing, DNA ligase is used in genetic engineering. Why do you think DNA ligase is used? I told you, it's like it does the glue activity. It basically makes the sticky ends join together properly, and it has a proper DNA, recombinant DNA being formed. That is the use of DNA ligase. So because it joins two DNA fragments by forming phosphodiester bonds between their sugar and phosphate backbones, helping to seal the foreign DNA into the vector. So we are taking the foreign DNA into the vector, and to seal that and to make phosphodiester bonds. Phosphodiester bonds between what? The bonds between the deoxynucleotides. Remember nucleotides, deoxynucleotides. So to make a bond between all of them, you need DNA ligase. Sticky ends helps, but then final work is done by DNA ligase itself to form a new recombinant plasmid out there, or basically for sealing the foreign DNA into the vector. It can be a plasmid, it can be anything else as well. Going ahead, chilled ethanol is used in DNA isolation. Now that is something new, that is something that we do not know. So first of all, my dear children, as I told, we have to take the gene of interest from our body, right? So in our cell there is RNA, there is DNA, there is this and that, so many things are there. So we treat it with enzymes. Okay, so we treat certain things with enzymes. So first of all, I want to brief you up. Since this topic is being there, I'll tell you about the entire process of isolation of DNA. In that way, that topic is also going to be completed. So suppose, my dear children, you have a plant cell. Okay, I will give you three scenarios. Okay, cell wall is there. Now suppose it's a plant cell. Plant cell, in the case of plant cell, the cell wall is made up of cellulose, which is why we need to first treat the cell with cellulase enzyme, such that it can break down the cell wall out there. Second thing, suppose I have, this is for plants. Suppose it's a fungi. In the case of fungi, the cell wall is made up of chitin. And for breaking into the cell of these, fungi, we need another enzyme which is known as chitinase. It is known as chitinase. Now suppose you need another thing. For example, you have bacteria. In the case of bacteria, my dear children, they have their cell wall made up of peptidoglycan. Their cell wall is made up of peptidoglycan. And for that, you need enzymes which has no resemblance to the name, and that is known as lysozyme. Remember, in our tears, in our saliva, we have lysozyme. These, which is why we say tears are very, very important. They're very, very useful out there, because these are these enzymes such that it can kill any bacteria which basically makes us itch our eyes and things like that. That's why we tear up. Okay, so that it's a defense mechanism of our body. The lysozyme basically breaks down the cell wall of the bacteria and kills it. Okay, so this is how you basically enter. Now once you enter, remember all these enzymes names, super important, you should know. Okay, now the next thing, once it enters in the nucleus, you can have the RNA, you can have the DNA, and you can have the protein. What do I need? I just need the DNA. So that means I have to break the other things down. So for breaking down the RNA, we have the enzyme RNase. We have the enzyme RNase. Okay, it can be broken down with the help of that. For proteins, we have protease. So all of it is treated very nicely with protease, RNase, everything, such that the RNA, protein, everything is gone off. And now we just have the DNA which is remaining. Now the DNA which is remaining, my dear children, it has to be isolated. How to do that? It has to be moved away from all the extra things which is there, which is still there. All of it which has been broken down, which is being cleaved, you have to separate it from all of it. So what do you do, my dear children? You are going to treat it with chilled ethanol. Okay, chilled ethanol. Remember that. Chilled ethanol is used for isolation of DNA. I have a GIF. I will be showing you that particular video GIF as well. Okay, so that is the use. Please remember all this information, super important. Now the smaller DNA fragments move farther in a gel slab. Why do you think the smaller DNA fragments? Because, my dear children, they are easy to move. They can easily move. And always remember, you have to tell extra things. For example, in gel electrophoresis chamber, as I told you, there is negative charged thing over here which is known as cathode, and over here you have anode. Always remember cat. In that way you can remember cat. C here, A here. Okay, and the things that are moved is DNA. DNA is negatively charged. This guy is also negatively charged. Anode is positively charged. So DNA moves like this. And also remember one super important thing, that is DNA does not move on the above surface of the gel. So here you call it gel electrophoresis. Why? Because all of it is happening in a gel which you call as agarose gel. You call it as agarose gel. It is made up of agar. So this thing you get from seaweed. You extract this from seaweed. Okay. Now this is all things happening in the gel. So do you think the DNA is moving on the surface of the gel? No. DNA shows something which is known as sieving effect. So basically, it is sweeping within the gel and moving. Always remember, it's not on the surface, it is within the gel. This can be an assertion reason question as well. Is this true? Is this false? You should know that. Okay, now the DNA is segregated, separated. How do you see it? You need a stain for visualizing it. You call this stain, that is also super important. It is known as ethidium bromide. Ethidium bromide is something that you need, and it gives you reddish orange color. Okay, when viewed under UV radiation. When viewed under UV radiation, it gives you reddish orange color. It is ethidium bromide, such that you can separate the DNA fragments. You can make it visible with the help of that. So chilled ethanol is for isolating the DNA, separating it, whereas this is for viewing the DNA on the gel electrophoresis chamber. Okay, so smaller DNA fragments move farther in the gel. How do you write it? Because during gel electrophoresis, smaller fragments face less resistance from the gel matrix and therefore migrate faster and farther towards the positive electrode, which you call as anode. Give an example for that. Okay, talk about that. You do not need to mention about ethidium bromide. I am telling you, I am explaining extra things because questions are you are gonna see questions, and I am basically explaining it priorly itself. Okay. Now, thermostable DNA polymerase is used in DNA polymerase chain reaction. Why? We all know that. Because, my dear children, DNA polymerase chain reaction happens at a very high temperature. Okay, at a very high temperature. And the most high temperature is 95 degrees Celsius. And in which step is that temperature? In denaturation. The first step, no, where we separate the DNA strands, that has the highest temperature. So for that, we need DNA polymerase, thermostable DNA polymerase. So it's not like DNA polymerase is put just during the replication process or extension process. It's just there throughout the process. Okay. So because the enzymes like Taq polymerase can withstand the high temperature used to denature DNA strands, allowing the PCR cycle to continue without the enzyme breakdown. So basically, that is why we use thermostable DNA polymerase or Taq polymerase. So always remember, the major step over here is denaturation. So usually I also thought that we need DNA polymerase for extension, right? So it won't be there, but it's there, and it is able to withstand this high temperature which is there in the denaturation step, that is 95 degrees Celsius, very hot. Okay, so these are the things that you need to know. And over here, this is chilled ethanol. As you can see, the DNA is getting separated. This is how gel electrophoresis looks. To making it visible, you need what? You need ethidium bromide. Under UV rays, it appears to be, I think, reddish orange in color. And over here, I have what? I have the DNA ligase, as I told. Ligase, the gluer. So over here, it is gluing things up. Okay, it makes the phosphodiester bond. So yes, that's all. Cool? Now, question number 9. Which of the following is not a feature of plasmid? Super important question. So you can't be asked as what are the features of plastids? Explain that. Sorry, not plastid, plasmid. Okay, plasmid. It's not plastid. Plastid is an organelle in the case of plant cell. This is plasmid. So what is not a feature of plasmid? It is extra chromosomal. It is circular structure. It is single stranded. It is independent. Shows independent replication. So my dear, suppose I take an E. coli. E. coli will have its own genetic material, okay, which is responsible for it to survive. It makes enough proteins and things like that for survival and everything. There is extra chromosomal material. Extra chromosomal means it's an extra thing. It's not the actual main guy. It's the extra chromosomal content right over here, my dear children. And this extra chromosomal content, as you can see, it is circular in shape and it has its own origin of replication. I told you. If it has origin of replication, it will do independent replication as well. It will replicate by itself. Okay. Now, my dear children, what is not the character that is being shown over here? Single stranded. No, it is not single stranded. Rather, it is double stranded, as you can see over here. So this is the thing that is odd one out. This is from model question paper 3, question number 10. So this is the features that you need to remember. It's extra chromosomal. It is circular structure. It shows independent replication, and it is act like a vector. It is going to act like a vector as well for basically the process of transformation to happen smoothly. So over here, as you can see, origin of replication is there, you have all this thing, but it is not single-stranded. Okay. Now, agrobacterium tumefactions is used as a vector for cloning genes in. Agrobacterium tumefactions is used as a vector for cloning genes in. This is MQP3, that is model question paper 3 question number 11. So you have seen 10 and 11 question is basically from this chapter. Okay, anyways, why do you think that this particular bacterium is used as a vector for cloning genes in first of all, in what? In plants. Okay, you use them for plants. Why, ma'am? Because, my dear children, I'll show you. Agrobacterium tumefaciens bacteria, right? It has a unique plasmid. You call this plasmid as TI plasmid. TI stands for tumor-inducing plasmid. Listen to me, do not read out anything as of now, okay? So this plasmid, which is very uniquely present in Agrobacterium tumefaciens, is known as TI plasmid. It is tumor-inducing. Why it is known as tumor-inducing? Because, my dear children, it has a segment which is known as tDNA, or you also call this as transfer DNA. So what usually happens is that, okay, what usually happens is that this particular plasmid, when the bacteria goes and affects a plant, it transmits the plasmid, the tumor-inducing plasmid, which has the tDNA, transfer DNA. The transfer DNA is responsible for inducing tumor in plants. It causes tumor in plants. Okay, now what if we use this machinery for our sake? What if we put our gene of interest over here, because anyway this is going to get transferred? So why not we remove this transfer DNA which causes problem and put our gene of interest, and then, my dear children, allow, anyway the bacteria is going to infect the plant, let it infect the plant, but while it infects, it is going to transfer my gene of interest, what I want. You get it? So basically, this is why I use Agrobacterium tumefaciens. So it is a usual organism that causes trouble in plants. Now I am going to use this machinery for this same good sake only, but it is again going to be for plants itself. So you have to remember the name of TI plasmid, that is tumor-inducing plasmid, and in this plasmid you have the segment which you call as tDNA, transfer DNA. Got it? Done? Okay? Now for animals, you have something different. For animals, what do you have? In the same question, I'll explain. For bacteria, what do you have? For viruses. So basically, for animals, I'll tell about animals, okay? For animals, we have retroviruses. You know HIV? HIV is what? A retrovirus. What does it do? It puts its genetic material into our body and makes our body produce its own proteins, things like that, right? So there are some retroviruses, but these retroviruses are going to be disarmed. Disarmed means basically, suppose I have the weapons, I take away the weapons, someone takes away my weapons, so I am disarmed now. I am not going to cause any trouble to anyone. Okay, I do not have the weapons. Similarly, the retroviruses, which usually causes problems, they are disarmed and they are inserted with the gene of interest. And now the retroviruses are going to be infecting the animals. So for animals, as vectors, you use retroviruses. In the case of plants, you use Agrobacterium tumefaciens. Please remember this. This is super important. Okay? I hope this is clear. Just give me a second, okay? Yeah, so that's all. Now we go ahead. So for bacteria and all, we studied about the heat shock treatment, this and that. We use another plasmid and treat it with that. Okay, for viruses, we do not need it. So let's go ahead. So now, my dear children, you have MQP3, Model Question Paper 3, question number 44. This is a 5-marker question and this is the easiest one. So genetic engineering is a core technique of biotechnology. In the background of this, complete the following table. So you have to tell what happens all over here. So gene electrophoresis, A, visualization of DNA. So basically, gel. So gel electrophoresis, in that, what do you use for visualization of DNA fragments? I just told you, you use ethidium bromide. What do you use? You use ethidium bromide. Now the next option is, over here the tool is DNA polymerase. What is this and what is this? So basically over here you have to name the tell the name of the process, and over here you have to tell the function. What is it happening in this particular process? So in gel electrophoresis, we are using ethidium bromide for visualization of DNA. Over here, it is PCR, okay, polymerase chain reaction, where we are going to use DNA polymerase, and this will be for what? Amplification of the DNA, for increasing the number of DNA strands out there. Now what is the next thing that you might know? You will not know this because as of now I have not covered it. Let's go ahead with the next one. Isolation of DNA dash precipitation of DNA. So for isolation of DNA, I am going to use something that is going to precipitate my DNA out. What is that something? It is going to be chilled ethanol. Okay, it is going to be chilled ethanol. Now over here you have this one, gene gun dash transfer of recombinant DNA. So my dear children, how do you basically, suppose I do not need vectors, okay, I do not need vectors such as agrobacterium tumefaciens, or I do not need retroviruses. What is the other way for making the cells competent, that is transforming the plant cells and animal cells? So you need to know two very important process. One is gene gun method and the other one is micro injection. Okay, gene gun method is also known as biolistics. It is also known as biolistics. So what do you exactly do over here? Here, suppose it's a plant cell. Oh, just a second. Suppose it's a plant cell. Now, plant cell, my dear children, has cell wall, which is why I cannot randomly insert the DNA of interest very easily, because it has a cell wall. I need to have a tougher approach to it. Suppose I have a gun. Okay, not the gun as you imagine, but a different way of gun. And this gun has bullets in the form of gold and tungsten. It is made up of gold and tungsten, my dear children, gold or tungsten. And over here, the DNA is being wrapped around it. Okay, not in the exact sense, but I'm telling you, it's coated with the DNA of our interest. Now, we put it inside the nucleus of the plant cell, which is why we call this as gene gun, and it is also infamously known as biolistics. Children usually forget about that. Now, in the case of animal cell, things are going to be quite different because they do not have cell walls. So it's very easy. So rather we use a needle for inserting the gene of interest directly into the nucleus. That is known as microinjection. These two processes are vectorless methods. Okay, these two processes comes under what? Vectorless methods of transforming the organism or transforming the cell out there. Okay, here we are not using any vectors. So gene gun method and also micro injection. Apart from that, there is one more method that is known as electroporation that we usually use in the case of bacteria and things like that. Electroporation is basically, I give electric shock such that it creates pores, and with the help of, through the pores, things can be inserted. That is one more thing. So here gene gun is given, dash transfer of recombinant DNA is there. So what is there? Over here, gene gun, dash transfer of recombinant DNA. So gold and tungsten balls would be there coated with the DNA. That would be the thing. So here is the entire thing. Just go through it. So gel electrophoresis, ethidium bromide is the equipment that you use for visualization of DNA fragments. Now over here I have what? Polymerase chain reaction. DNA polymerase or Taq polymerase is what we use for amplification of the DNA. Now over here what do you have? You have the gene gun. Tungsten or gold particles coated with the DNA is being used, and it is for transfer of recombinant DNA. And last but not the least, you have isolation of DNA. For that you need chilled ethanol, which basically causes precipitation of the DNA. And these are the things that you need to keep in your head. Done? This is model question paper 3 question number 44, and with that I think we have covered all the model question paper questions done for the year 2025-26 of this particular chapter. Now we are going ahead with what? The revised question bank questions, the PYQs and MQPs. So I have tagged the questions that I have taken from the question bank. I have not tagged the previous year questions and MQPs because it's a huge lot of work and I don't think it's needed as well. So let's see. Okay. So agrobacterium tumefaciens transform normal cells into a tumor by delivering a piece of DNA known as. What is the DNA that is being delivered? We discussed, it is known as transfer DNA. So question bank question number 14. So in the question bank, revised question bank, they have tagged the question as easy, average, difficult. So I have taken the average and difficult questions over here. Easy ones I have not taken. Okay. So while we do marathon during the examination, at that time we can take the easier questions as well. Cool? And if you have any doubts at any point, put it down in the comment section. I am here to solve that. Fine. So this is tDNA or transfer DNA. This is the one that causes tumor in the case of plants. Now, guess. Which of the following is the correct order of processing of PCR? Question number 19. So since we have studied all the things now, it's very easy for us. Remember, what are the correct order of processing of PCR? First, you need to denature, then you anneal the primers. What are primers? Deoxyribonucleotides, okay, DNA. Deoxyribonucleic. Okay, fine. So we do annealing, and then what do? We do extension with the help of Taq polymerase. So first denaturation, then annealing, then extension. Your answer would be C. While doing these type of questions, always recheck. Sometimes we get things wrong. In your head it is this, but in your while you write it becomes B option. So please don't do that. Read the questions carefully and then only do it. Okay, so it is C option, and this is the whole thing. Highest temperature is needed over here. This is why you need thermostable DNA polymerase as well, because high temperature is maintained throughout the process, but in denaturation 95 degrees Celsius. Over here it is little less in annealing, and over here again it is 72 to 75 degrees Celsius. Okay. Now, 14th statement, this is from question number 21 of question bank. Okay, statement 1 says sticky ends are resulted due to the action of restriction enzymes. Yes, that's how sticky ends are got. Yes. Now, stickiness facilitates the action of DNA polymerase. This is why I told you should know the name of the enzyme. Do you think the stickiness facilitates the action of DNA polymerase? DNA polymerase extends the DNA. No. It is DNA ligase. Stickiness facilitates the action of DNA ligase, which has to join the fragments out there. So I'll say statement 1 is true, statement 2 is not. Statement 1 is true, statement 2 is what? False. So your answer for this question becomes A. Cool. Okay, we discussed all of it earlier itself. Again, we do not need to do that. But please remember, you need to read the question properly. So whenever you get a question paper, be confident, be do not overconfident. Okay, be very careful. Okay, and do not be tense too much. So there are certain things. So you have to be balanced. Be cool, read the question carefully, read it again, read the answer as well. Okay, the purpose of stirred tank bioreactor. Now that is something new. Stirred tank bioreactor, have you seen the picture? Well, the answer for this question is D, availability of oxygen throughout the process, because I want to show you the picture. This is how it looks. Okay, you might have to draw this also. The picture of this also has been asked for you to draw, so you should know how to draw it. My dear children, you are asked what is the purpose of stirred tank bioreactor. So as you can see, there are some stirrers over here which moves. And why does it move? First of all, it moves because it wants to, what is this? First of all, so bioreactor is something where the microorganisms are growing. And why are we making them grow? Because they have our gene of interest. So when they grow, they basically are amplifying the gene of interest as well. Right, so that is the process. So for doing it in large scale, we have bioreactors. Now there is one bioreactor which is known as stirred tank bioreactor, and the other one is sparged bioreactor. That is different. In the sparged one, you have air bubbles flowing in through that. That is totally different. Now this is totally different. In stirred tank bioreactor, you have stirrers like that. So first function of it is to basically provide the nutrients equally to all the cells out there. We give food, okay, food in the form of nutrients and stuff like that. So that should be mixed. Apart from that, the major function is to make sure that there is availability of oxygen throughout the process. Remember, here you need oxygen. These are aerobic bacteria. These are not anaerobic. These are aerobic bacteria. They need oxygen, and to make availability of oxygen throughout properly, we need stirred tank bioreactors. Now over here, there are some important things, such as you would need motor, such that it can move around. You need acid and base for pH control. So over here, this is a pH control thing. Okay, this is stream for sterilization. Here you have foam breaker. Okay, if there is too much foam being formed, my dear, that will be a big problem, which is why you have a foam breaker. And this is the flat bladed impeller. You have the culture broth. That means the bacteria as well as the nutrient medium which is there. So please remember, you should know about all of them. Motor, foam breaker on this side, flat bed. This is foam breaker at the top, because foam usually forms at the top and you want to break that up. You can be asked, what is this in the question paper? A, B, C, D. So you should know about all of it. Okay, and here you have the sterile air entry as well. Sterile air, it should not be mixed with anything else. So the purpose of stirred tank bioreactor is providing anaerobic condition? No. Aerobic condition, you need air. Purification of the product? No. Addition and proper mixing of the preservative? No. Preservative and everything you add later on once you extract the product. As of now, we are just increasing the number of bacteria, and then later on we will take the proteins or whatever gene of whatever product we need, we will take it with the help of downstream processing. That is the next step. Okay, this is just a large scale production. So over here you just have one answer, and that is to make the availability of oxygen throughout the process. Now, next question back question number 16. Identify the correct statement. Hint 2 cut the DNA at specific sequence of 6 base pairs. Hint 2 was the first restriction enzyme, and does it cut at the specific sequence of 6 base pairs? Yes, ma'am. So this is the correct statement, first one itself. But let's see. Microinjection is a technique of introduction of rDNA into the plant cells. Recombinant DNA RV. Are we putting inside the plant cell with the help of microinjection? No. Injection you for animal cell. Always remember about your injection. Okay, so that's the best way to remember. So this is wrong. Biolistics is a technique of introduction of rDNA into an animal cell. I do not know what is biolistics. Arey baba is gene gun, and gene gun method is not for animals, it's for plants. Retroviruses are commonly used cloning vectors in the case of plants. No, I told you retroviruses are for animals. Okay, so while you use vector, this is a vector for animals. Vectorless method is microinjection. Okay, so only the correct statement for this question would be A. Children, don't you agree that while we are going ahead, we are we know things a lot more better now? Yes? Do you get that confidence? If yes, let me know. Do you love the session that is happening right there? If you are liking it, let me know that as well in the comment sections. Okay, I'll be really motivated. So this is how you can go through the entire thing. This is, as I said, it's for you to refer. This was an MCQ question. I just give you an explanation over here if at all you want to read it later on. Cool? Now question number 17. Statement number 1. Plasmid DNA acts as vector to transfer the piece of DNA attached to it. So plasmid DNA which is there, do you think it acts as a vector to transfer the gene of interest that I am attaching over here? Yes, ma'am, this is correct. Vector DNA acts as plasmid to transfer the piece of DNA attached to it. Vector DNA acts as plasmid. No, plasmid itself is a vector DNA. So what is this statement? This statement does not make any sense, which is why this is wrong. Plasmid DNA acts as vector to transfer the piece of DNA attached to it. Vector DNA does not act as plasmid. There's no need for that, okay? So basically the statement does not make any sense, which is why only statement 1 is true, whereas statement 2 is false. Okay? Cool. Let's go ahead. Now, we are entering 5 marker questions, and let me tell you. So in the question bank, you have the previous year questions also. In the question bank, you have the previous year questions as well. So from now on, it will be the previous year questions that I am going to discuss. I have not mentioned the year because it's there in the question. Same question is there in the question bank as well. Okay, so it will be too much cumbersome. You will see this question here, you will see that question there. So that's why there's just simple thing that I have included over here. Question number 18, which is a 5 marker question, says that given below are the diagrams of plasmid A and B. Observe meticulously and answer the questions that follows. Now these are the things that you probably might have not studied until now. Okay, which plasmid is or are you select for cloning and why? So you have 2 plasmid. One is your PBr 322 mentioned over here. Another one is plasmid B. Okay, over here what do you have? What do you have as a unique thing? This is LAG-Z gene. So LAG-Z gene basically is there for coding for an enzyme which is known as beta-galactosidase. Okay, this particular plasmid is known for this particular gene, that is LAG-Z gene, that codes for an enzyme beta-galactosidase. Remember in lag copron we studied LAG-Z, YA, right? So Z is for beta-galactosidase. What does it do? So suppose I put a chromogenic, a colorful substrate into the medium that is having this plasmid, such as X-gal. X-gal is the chromogenic material, the one that gives color. Suppose I put it and I have the LAG-Z gene. LAG-Z gene will produce the enzyme, and the enzyme will break this particular thing, which is why, okay, which is why the colonies would be giving off a color which is blue color. Okay, it will be giving off a color which is blue color. Just give me a second, okay? Yeah, it will be giving a color that is known as blue color. Okay, and suppose over here I am inserting something with the help of BAM H1. I am inserting something over here. Do you think it will still produce beta-galactosidase? The answer is no. It is not going to produce beta-galactosidase. And if beta-galactosidase is not being produced, my dear children, do you think the chromogenic substance which is there, it can be there is something happening onto it? No, which is why there will be white color. There will be white color. Okay, so blue color, if at all, it's normal. White color, if at all, recombination has taken place. So that is the thing, general idea for you. Okay, now we go ahead and we see this question. Which plasmid or is or are you select for cloning and why? Among them, which is the one that I may select, I will select this one. Why, ma'am? Because it's easier. Either I can see blue color colonies, either I can see white color colonies. If it is white, I get to understand that recombination has taken place. It's very easy. But if I do this, I have to do so many plating. And you will get to understand more about it when I solve the whole question. So don't be worried if at all you do not understand it at this particular point of time. Now what is insertional inactivation? We discussed about this. Insertional inactivation is basically, suppose a restriction site is somewhere in a gene coding for something else. Now when I put my gene of interest over here, okay, what happens? The normal gene which was there, it stops working, it becomes inactivated. This is known as insertional inactivation. So you either can give an example of this where if you cut with BAM-H1 and then probably insert a gene of interest, then it won't be tetracycline resistant anymore. Or you can give an example of this, if at all you insert something over here, LAC-Z gene won't be producing beta-carotenosidase, and which is why there won't be color formation as well. You can explain in whatever ways you can. So that answer is given over here. It means the gene stops working because a foreign DNA is inserted into it. Example, in plasmid B, the LAC-Z gene stopped producing the enzyme beta-carotenosidase after the insertion of what? Foreign DNA. Now, will the number of culture plating you should make is same or different to select recombinant if insertional inactivation is possible? Comment. So now, they are just asking you this one very important thing. What is the way how you can find the recombinants here and what is the way that you can find recombinants here? Which one is more troublesome? Which one takes more time? Which one has more culture plating? For in simple words, which one does take more efforts? Easy. So I'll tell you how you can solve this problem. So my dear children, I have two plasmids. This is plasmid B. This is plasmid A. Plasmid B, my dear children, has the LAC-Z gene. I'm explaining everything, okay? Which codes for beta-galactosidase, which basically reacts with any chromogenic substance. That means something that can give color. And over here it is X-gal, okay, the material is known as X-gal, and it gives blue color in general. Suppose over here I am putting my gene of interest. In this case, none of it is going to happen, which is why the bacteria that has this particular plasmid, okay, will show white colonies, will have white colonies. So in a plate, suppose I put the chromogenic substance, that is X-gal. I have two bacteria over here. One set of bacteria giving blue color, and other one basically giving rise to white colonies. What do I understand from this? I understand that these bacteria has my DNA, or basically has the recombinant DNA, whereas this ones are non-recombinants. I just need one plating to be done, and I can take this bacteria then and I can use it for my purposes, because these are the ones that gives white colonies. So these are the recombinants out there. Agreed? Only one plating done. It's very simple in this particular case. Now if it is this plasmid A, my dear children, where we use PBr 322, you have ampicillin resistant gene over here and you have tetracycline resistant gene over here. Suppose I make a cut through, so in this case it is resistant to both ampicillin as well as, I'll write it, resistant to both ampicillin and tetracycline, right? Now over here with the help of BAM-H1, I am going to insert something right over here. in the tetracycline resistant site. Okay. Now my dear children, the first plating that I'll be doing, okay, will be, I'll basically try to grow the E. coli in a medium that has ampicillin. Now the E. coli that has taken up the plasmid, okay, that has taken up the plasmid, which is this, it will be surviving. Why? Because it is having ampicillin resistant gene very active over here. It is having the ampicillin resistant gene very active over here. If we see the tetracycline one, that is not active. Why? Because I have inserted this blue color thing is what my inserted thing. So it is not tetracycline active, it is ampicillin active. So it can survive in ampicillin medium. Okay. There can be another E. coli which is having a plasmid that does not have it. That does not have this particular plasmid. It has its own, but it does not have these particular plasmids. So with the help of this particular culture plating, I am able to understand the one that has become, that are transformants. At the end of culture plating 1, I am able to identify the transformants, the one that have taken up my interest of plasmid. Agreed? Now over here, suppose there can be one more thing. There can be one more thing. An E. coli that has both ampicillin as well as tetracycline resistance. Because over here probably the gene of interest was not being put inside properly, which is why there will be some plasmid that will be like original. In original case, what was there? One side was ampicillin resistant and the other one was tetracycline resistant. The original one was like this, right? So the original one can be also be there. So basically, E. coli can take up plasmids which has the recombinant DNA. E. coli can just take up plasmids which is new, and E. coli may not take up any plasmid at all. That has the 3 conditions. So basically, while I try to grow in ampicillin media, at least I can rule out this one which do not have the plasmid of my interest. So the non-transformants, okay, the non-transformants are removed when I grow it in ampicillin. I have 2 of them now. I just have 2 type of things now. Okay, this is the recombinant, this is the non-recombinant. Non-transformant and non-recombinant is different. Transformant means I have not taken up anything new. Okay, non-transformant means I have not taken anything new. These 2 guys are the transformants. Now out of these 2, one is the recombinant, other one is not. That means the one is the one that has the gene of interest, other one does not have the gene of interest. For that, I need to grow them in another media that has tetracycline. When I grow it in tetracycline, my dear children, I have these 2 guys, right? Both of them will have ampicillin resistant gene. One will be tetracycline resistant, another has gene of interest, other has my gene of interest, which is why it is not tetracycline resistant. So which one will survive? This one will survive, this one would not. So the recombinants, my dear children, the recombinants will not grow, and the non-recombinants which are there, they are going to grow. Non-recombinants will grow. And with that, I get to understand that this is what I need. These are the cells that I will be needing. Okay? And the whole thing is gone. I cannot bring it back. So basically for that you need 2 mediums. Okay, for PBR 322 you need 2 mediums. First is to understand what is transformant. Second is to understand which one is recombinant, which is why it takes a lot of your time. Now that is the question given to you. Will the number of culture plating you should make is the same or different to select recombinants if insertional inactivation is possible in both of them? No, it is not going to be the same. The number of plates will be different for both plasmids. For plasmid A needs 2 plates, one with ampicillin for finding the transformants and one with tetracycline to finding the recombinants. Whereas for plasmid B, the needs only 1 plate with X-gal, white for recombinants and blue for non-recombinants. Okay, so hence plasmid B will be lot more better because things are going to be faster. Now over here also the first question, which plasmid is or are would you select for cloning and why? You will select this because it is easy to find recombinants in them. You just need 1 plating being done. The white colonies are recombinants, the blue colonies are non-recombinants. Easy. Now this question usually cracks up a lot of minds, which is why I explained you, but I still know that a lot of you people might be sitting like this. So I want you to pause the video and retry it once. I'm telling you, it's super easy once you try once more. Okay? Now study the diagram given below and answer the question that follows. What is EcoR1? EcoR1 is basically a restriction enzyme. Now you have to explain a bit more. Where do you get E from? So eco is basically from Escherichia coli, that is the name of the bacteria. So the first letter is E, CO is the two letters of the species name. Now R1, so R is basically the name of the strain. So RY13 is the name of the strain from where you took R. Okay, now 1 because it was the first enzymes that was discovered from the E. coli. Okay, first restriction enzyme that was discovered from the E. coli, which is why EcoR1. Now in the order of discovery you can change 1, 2, 3 and things like that. So EcoR1 you have to explain, it's a bacteria, is a EcoR1 is what? A restriction enzyme isolated from the bacteria Escherichia coli. It recognizes the specific palindromic sequence. This is the sequence and it cuts between G and A and producing what? Sticky ends. And please mention about how do you get the name also. So E from the first letter of the genus name, 2 letters of the species name, R from the strain and 1 from the order of discovery. Okay, now next question, how is the action of exonuclease different from that of endonuclease? So ma'am, until now we studied about restriction enzymes, but you didn't tell that there are 2 types. There are actually 2 types, one is endonucleases and the other one is exonucleases. Endo means they are going to cut within the DNA strand. Okay, they're going to find the sequence, they're going to cut within the DNA strand. Exo means they're going to cut towards the ends, not in the middle, towards the end. So how do you differentiate? So exonuclease cuts DNA from the ends of the strand, endo cuts the DNA within the strand itself, removes nucleotides one by one from the terminal ends, exonucleases, whereas breaks down DNA specific recognition sequences. Give an example over here, you do not have an example in your NCERT, so go ahead with the example of at least the endonucleases, that is EcoR1, HIND2, HIND3, whatever is there. Okay, so it cuts the DNA from at the end, it cuts from within, and over here it removes nucleotides from the terminal ends. Here you have specific recognition sites. Okay, all of it. Next question, how are sticky ends formed on a DNA strand? Why are they so called? So how are they formed? When sticky ends are formed when a restriction enzyme such as EcoR1 cuts the DNA staggerly between G and A. So you have to give a picture and then explain. So please go ahead with the picture. You have GAATTC, you have GAATTC. Make a cut over here, make a cut over here. You get an L-shaped thing which is still left. Okay, you will get an L-shaped here which is attached to this. Again, you will get an L-shaped thing which is attached over here. As you can see, these L-shaped thing which are joined together, basically they are sticky ends, they are joined together to form a recombinant DNA. Okay, so suppose this is supposed to be a vector DNA, that is your plasmid DNA, and this will be your foreign DNA, and then you're making, so this is just one side. Similarly, the same thing will be happening on other side of the foreign DNA. Same, suppose this is the foreign DNA, on the other end also you have the same GAATTC, and the plasmid will also have, there it gets sandwiched. Okay, so we initially spoke about it while we spoke about this picture. Just a second. So I tell you the picture, it will be more easy for you to understand then. Remember we studied about this picture. So in the target gene, but both the ends we are cutting. Over here you're showing just one end and you're showing the one end of the plasmid. That is why you might not be able to understand, but I hope by seeing this picture you might get an understanding of things. So where are we? We are over here. Yes. So you have to talk about it as well. This produces single-stranded overhangs. Yes, single-stranded overhangs, which you call as sticky ends. Okay, they are called sticky ends because these ends can easily pair or form hydrogen bonds with the complementary bases of another DNA fragments. Okay, so you can go ahead with that, and you can say that this is used for joining with the vector DNA. DNA ligase does the other job and everything. Make a diagram, explain it properly. It will be more easy for you to understand as well. Cool. It's a 2-marker question. Now write 3 vector-free techniques that can be utilized to transfer the recombinant DNA into a ready host cell. Vector-free, microinjection, biolistics, electroporation. So explain that. So microinjection, directly injecting recombinant DNA into nucleus of an animal cell using a fine needle. Bialistic or gene gun, high velocity tungsten or gold particles coated with DNA are shot into the plant cells. And electroporation shot into where? The nucleus, okay? Electric pulses create temporary pores in the cell membrane allowing DNA to enter. So that is also a vector-free method. Now, Agrobacterium tumefaciens acts as natural vector for cloning genes in plants. Why natural vector? Because it usually infects the plants. As I told, it causes tumor, but now we are removing the tDNA part, the transfer DNA part, and we are putting our gene of interest. So again, you have to explain that. Agrobacterium tumefaciens is a soil bacterium that naturally causes tumors, crown galls, that is what you call, okay, in the plants. It carries TI, plasmid, that is tumor-inducing plasmid, that transfers a segment of DNA called tDNA into the plant cell. In genetic engineering, this TI plasmid is modified to remove the tumor-causing genes and used to transfer desirable genes into the plants. Hence, it is a natural genetic engineer or natural vector to the transfer of things in the plants. Natural vector for animals, it is retroviruses, okay, disarmed retroviruses. Now here comes the question number 21. Write a label diagrammatic representation of recombinant DNA technology. Now this is given in your NCERT. And you have to go as such. So foreign DNA, you have to tell how things are getting processed. So foreign DNA is there. You take the enzyme, restriction enzyme, and you cut it. Similarly, you use the same enzyme for cutting the vector DNA. And now, my dear children, you're using ligase to join the foreign DNA to the plasmid. Now the plasmid is made to enter into the cells. This is known as transformation. Now the cells will divide, and obviously what will happen? The gene of interest is also being replicated. Now in between that, you have lots of other processes, just gel electrophoresis, this and that. So what you can do is basically mention that as well. Okay, so suppose over here you made the cut. Okay, you made the cut, and then you can just put an arrow and say gel electrophoresis is done, and then PCR method is being utilized, and then, my dear children, basically what is done? Yeah, PCR method, and then the number of things is being increased, then you use ligases to join them, and the whole process goes off. But if at all you want, you can keep it as simple as possible and draw it like this also. You will get your marks like this as well, because in your NCERT this is written as diagrammatic representation of recombinant DNA technology. So that would also be fine. So don't use too much of your brain, just do this, it will be more than enough. If you want, you can write it down. Instead of doing this picture, you can write it down that for separation of DNA you have gel electrophoresis, and then for amplification you have PCR as well. Mention that, okay? And then downstream processing is also there. What is that? We'll study over here. This is the last question for the session, my dear children. Listen to me very, very carefully. So identify, just a second, okay? Yeah, briefly explain the steps of recombinant DNA technology. So what is the first step? Isolation of DNA. What happens over here? We are going to get the DNA from the donor or the source. Okay, so enzymes like lysozymes used for bacteria, cellulase for plants, and chitinase for what? Fungi. So basically it breaks down the cell wall and gets into it. Now you use RNAs and protease to digest the RNA and protein, and whatever is left is the DNA that is being precipitated with the help of chilled ethanol. Talk about that. Okay, now cutting of DNA using restriction enzymes. The next step, you cut it. So restriction endonucleases cut the donor and the vector DNA at specific recognition sites. Remember, both donor and the vector DNA are cut with the same restriction enzyme, producing what? Sticky ends that can join later. Now separation of DNA fragments, gel electrophoresis. So basically now we are going to separate them. As I said, mention about cathode, anode, tell that DNA is negatively charged because it is why it moves towards the anode, and always remember smaller fragments move faster, and it shows sweeping effect. It does not go from on the top. Okay, now insertion of desired DNA into the vector. Now once that has been done, you are going to insert the desired DNA into the vector, my dear children. That is our plasmid. Now introduction of recombinant DNA into the host. Now you put into the host, you can either use vectors or you can use vectorless method. Now selection of recombinants. How do you do that? With the help of selectable markers. Well, I didn't tell you the antibiotic resistant gene or anything are there. All the genes which are there, which is there for selecting, you call them selectable markers. What do you call them? You call them selectable markers. So selection of the recombinants with the help of selectable markers, and the expression of gene. Last, the host cell machinery expresses the inserted gene. Whatever gene is there, it will express the protein. Okay, whatever protein has been made, that has to be taken down as well. That is done through downstream processing. So after the biosynthetic stage, the product undergoes downstream processing, which includes separation, purification, formulation with preservatives, clinical trials, and quality testing before it's ready for marketing. So these are the things. So expression of the gene is also one thing. It will produce some protein, and that protein has to be separated, okay, and then it has to be preserved. All of it comes under downstream processing. Separation and purification comes under the downstream processing. That is the last step. Okay, cool. So expression of genes. Before that, obviously selection of recombinants, and then, my dear children, you can also have the large scale production. Okay, the large scale production using what? bioreactors, using bioreactors, and then expression of gene can come. Once you have produced enough quantity, then you do expression of gene thing also. That means you got enough bacteria, now bacteria will be producing enough proteins, that is expression of genes. And then the other processes, separating all this thing from the bacterial cells and the content, everything comes under downstream processing. And over here, please mention about PCR mechanism also, amplification of DNA also, it is also super important. Okay. Separation of DNA fragments, then basically you can say separation of DNA fragments, then you can talk about PCR, amplification, and then insertion of desired DNA into the vector. Okay. Now there is a question like what are the features of ideal plasmid? So you should know that it should be having the ORI site. ORI site is responsible for how many cycles of replication can happen. So a plasmid that has enough replication cycle is being taken up, and it should have selectable markers. Okay, plasmid should have selectable markers, otherwise the whole process would not be of any use. And it should have much cloning sites. Cloning sites as in the restriction enzymes which are there, they should be there, their site should be there. Always remember, for one restriction enzyme there should be one site only. For example, in PBr 322 there is only one site for BAMH1, right? Why not multiple sites? Because if it has multiple sites, it will cause multiple fragments, and that will be of no use. We do not want that. We want unique. So it should have multiple cloning sites for multiple enzymes, that is there. But if at all I'm talking about one enzyme, it should have just a unique site. So in PBr 322, I have 8 cloning sites, that is fine. But each of them has only one site. That is also there, please remember that. So you need ORI site, you need what? You need selectable markers and also enough cloning sites. So these are the ideal features of plasmid. And with that, we end this. Now tell me, was this video helpful? Were you able to understand the concepts also? I went a little bit longer because this chapter was not explained by me in this channel for this particular day, which is why I thought I should be explaining it as well. If at all you think it was good, please let me know and please share it with your friends. This video is going to be super important for you. I'm telling you again, please watch it completely. If at all you're just going ahead and seeing what's there in the end. And my dear children, that's all. Lots of love to you. Take care of yourself. Wishing you all the best. You're going to absolutely rock the examination. See ya from Jasmine Ma'am.